Hello, good day, everyone. Welcome to attend today's Global Engage webinar. Today's speaker is Dr. Gianni Panagiotou, who is going to share with us about his fabulous research. I'm Jessie Zhang, the local customer account manager from Novagen Europe. It is our great pleasure to be sponsoring such a prestigious event. And before Gianni starts his talk, I would like to give you a very brief introduction to Novagen Europe. Novagen was first founded in 2011 and has been growing rapidly since then. Our company Novagen's mission is to improve research, clinical research, diagnostic, and human healthcare. As you can see through the slides here, our service covers the main aspects of NGS with a wide variety of applications. Our European Sequencing Center is based on the Cambridge Science Park in the UK. Here we have our state-of-the-art lab equipped with the latest Illumina sequencing machine, NovaSeq 6000. We provide sequencing services with fast turnaround time and high quality data. We also have our super computing cluster, so we can offer bioinformatics, analysis, and store your data in the UK. As you can see, Novagen is a global company. We can offer services across Asia and the US, and also, of course, Europe. With around 2,000 employees and serving over 5,600 customers from universities, biotech companies, and hospitals, we can provide sequencing for the smallest or the largest projects. We provide a full range of sequencing services that covers a wide range of applications from human genome, transcriptome, microbial genome, epigenome, plant and animal genome. For microbiome study, Novagen provides different solutions for microbial researchers, including whole genome resequencing and de novo sequencing for single species, as well as amplicon-based metagenomic sequencing and shotgun metagenomic sequencing for microbial community study. There are different choices for NGS service providers, but there are a lot of good reasons why you should trust Novagen as your sequencing partner. The top two reasons would focus on data quality and fast turnaround time. Novagen delivers unsurpassed data quality, which exceeds Illumina standard and is ready for paper publication. Speed is another critical factor for us to win the business. We are capable of sequencing thousands of samples per day. With multiple NovaSeq at our UK lab, we can sequence equivalent to 280,000 genomes or 25 billion microbial genomes per year. The sample size of microbial services could be 10,000 or just one sample. We are very proud of our record in the scientific community. We have been cited in over 670 research papers with a total impact factor greater than 4.930. We are also driving the NGS industry forward with over 37 NGS-related patents. Our technology is to helping make the NGS process more efficient and effective, all the while improving the quality of the data we deliver. So the Novagen community keeps on growing. Our expert sales team are located across Europe and are ready to assist you for your next project. We can help you achieve your research goals. If you'd like to get in touch, please leave your inquiry on our company website or send an email to info@novagen-europe.com. One of our lovely sales clicks at your location will be in touch to answer any of your questions. With that, I'd like to thank you for your attention, and I would like now to introduce today's speaker, Dr. Gianni Panagiotou. Gianni is the head of the System Biology and Bioinformatics Unit at HKI in Germany. He's also associate professor from University of Hong Kong. The research focus of him is to use artificial intelligence method to identify signatures of microbiome disbalance that allows for early detection of disease and to shape and control microbial consortia to create beneficial effects in patients with chronic disease. They are looking to understand the interactions between good bacteria and fungi and how these interactions can prevent systematic infections from opportunistic fungal pathogens, and are developing new tools for diagnosis and patient stratification in sepsis. I have worked with Gianni now for over 3 years at Norwegian, and I'm really looking forward to your presentation, Gianni. Now over to you.
Okay, thank you very much, Jessie, for the very nice introduction, and I can only really say and confirm that the quality of Novogen is really amazing in all the projects that we have worked together, and of course it's a great pleasure for me to be part of this seminar that you are organizing and share some of the data that we have and some of the studies that we did on microbiome. And actually, my focus today will be more on the translational potential of the microbiome. I hope that in the next 20-30 minutes, I will convince you that actually microbiome-based interventions are coming closer and closer to be applied in a clinical setting. Of course, for this particular audience here, I don't need to say too much about the importance of the microbiome. Is one of the hottest topics in recent years, with really tremendous productivity from the scientists that are involved in this field. And with productivity, I mainly refer to publications. If you look just 10 years ago, probably someone that was working on microbiome, he could read all the papers that were published that year. I mean, it was, I think, if I can see here, well, it's 160 papers on microbiome. If we look what is the situation today, last year it was around 9,000 papers. Probably it will be the same today. So for this year, so for the ones that are actually going to stay in this seminar series, probably by the end of the day, you will already be behind in literature. There will be many papers that will be published and will not have the opportunity to actually go back and see what was about. So the obvious question is, what have we learned from all these thousands of papers that are published every year? I think the first thing that we learned, it was really in contrast to what we learned when we sequenced the human genome. When we sequenced the human genome, what we found out is that we are all more or less the same. So we share 99.9% of our DNA. But when we sequenced the human metagenome, we realized that we are less similar in microbiome composition than we probably thought. And this low similarity is something that we should always keep in mind when we are designing interventions and when we are really considering of using the microbiome either as a diagnostic tool or as a therapeutic tool. Another thing that we also learn is that everything that we do can actually influence the composition of the microbiome. It's not only that we have a unique signature, but we have a signature that is also easily modified. And I think it was an interesting study that we did a couple of years ago, where we could actually see that the microbiome, the skin microbiome of an individual can really reflect even things like using the metro, which metro line this person used, whether it used it in the morning or in the afternoon. This signature was really reflected on his skin microbiome. Of course, you can say that there are not many clinical applications of this. If you really want to know if someone used the metro, you can simply ask the person. But at the same time, it also opened another range of possibilities and opportunities. And many researchers started working on using the microbiome actually as trace evidence in forensics, and not only research teams, but also the team of Horatio in CSI Miami. He used once the microbiome to solve a murder case. So even if you don't really trust papers that are published in Cell Reports, as long as Horatio is using it in CSI Miami, it must be working pretty well, right? Other things that we learned with all these studies on microbiome is that there is a clear association between this balance in the human gut and the development of diseases. This is not entirely new, I have to admit. Hippocrates, the ancient Greek doctor, had actually claimed 2,500 years ago that all diseases begin in the gut. We don't know how people really reacted into this very strong statement back then. I mean, there are always people in denial. But what we know today, with the help of next generation sequencing, is that actually this is to a very large degree true. It is, excuse me, but I don't know why there is a problem with my, yeah. So with the use of next generation sequencing, we have actually sequenced a lot of metagenomes, and with the development of machine learning algorithms, we have managed to really showcase that changes in the microbiome composition are indeed have a causal impact on the development of different diseases. And besides that, we have developed a lot of mice models, very nice mice models, either germ-free mice or chnotobiotic mice, that can actually help us to prove this causality. So where are we now? I mean, we have learned all these things. What is left for us? I think we are really at the exciting point that we can actually rationally manipulate the gut microbiome. And there are many different ways, as I said, that we can actually manipulate the composition in the gut. It could be through diet. This diet is in the driver's seat of microbiome changes. We know that already from 2014, when Peter Turnbaugh actually published his study in Nature, where he had two groups of people, either with animal-based diet or plant-based diet, and he could see that actually there was a very rapid response of the gut microbiome on diet. But we know also that exercise can change your microbiome. Probiotics, postbiotics, symbiotics, you name it, all can have an influence on your microbiome composition. And we are even reaching the point that we can make very precise manipulations of the gut microbiome composition by removing pathogenic species. And there is a Danish company with this very interesting name, Sniper, that is pioneering these approaches using CRISPR methods to actually remove selectively bacteria from the gut. So this is where we are, and I will share with you a couple of studies where we actually try to do this, try to manipulate the gut microbiome in order to influence particular phenotypes. So the first case study that I will share with you is on cancer. And the role of microbes on cancer is not new. We knew that, for example, Helicobacter pylori is involved in gastric cancer development, but we didn't know a lot whether microbes that we have in our gut or in gastrointestinal tract are really involved in the development of extraintestinal tumors. So we decided to test whether this could be the case using hepatocellular carcinoma as a case study. And we designed for this a very novel probiotic mixture using probiotics that are already in the market. And we had a reason why we did that. And the reason was that we were hoping that if actually it's working, if we can actually manipulate or regulate the immune system of the host by this probiotic mixture, that then it would be easier to transfer it in the clinic, something that we actually did. And now there are clinical trials with this probiotic mixture in liver cancer patients. But as in brief, what we actually did is that we had 4 groups of mice, one treated with a normal diet and then injecting the tumor. Then we had the second group that was having the tumor injection, and it was treated with cisplatin, an anti-cancer drug. And then we had the 2 probiotic mixtures. In one case, we gave the probiotic a week before the tumor injection in order to prepare the immune system. I don't know if actually you can see my cursor, but in the figure on the right, we observed really a fantastic improvement in the mice response. So the tumor size really shrinked in the case of probiotics, more than 40%. And that was really remarkable. With no other treatment, by just using this probiotic mixture, we could see a great improvement in the phenotype. And of course, the next step was to really try to understand the mechanism, how this was really possible. I will not go through all the details, but we had to measure a lot of cytokines, a lot of immune cells. I would say this is one of the very first studies that linked specific bacteria species with the immune landscape in mice. And what we observed was that actually the Th17 cells, the abundance of Th17 cells in the tumor correlate positively with the tumor volume. So we guessed that probably Th17 distribution is a key factor for modulating the cancer development in this mice model. So we then started looking into every single organ of mice to see where the probiotics were actually having most of the effect. And you can see that what we found out was that actually the use of probiotics is reducing the abundance of Th17 cells in the small intestine and in the blood. So basically, it inhibits the recruitment in the tumor of Th17 cells through the blood circulation from the small intestine. And then going further, we look, of course, into shotgun metagenomic analysis of our different groups, and we could pinpoint specific driver species of this phenotype, because our assumption is actually that the probiotic mixture that we added, it's not directly influencing the immune cell landscape, but it is actually reshaping the microenvironment in the gut, and different species with different abilities to produce anti-inflammatory metabolites are taking over and have this impact. And as I said, now this is in clinical trials in combination to see whether in combination with anti-cancer treatment, we can see further improvement. And actually, right after this study and a couple of years later, I think it was in 2018, we had also the first human studies, the 2 papers that were published in Science, one in melanoma and one in lung cancer, where they could demonstrate that whether you are going to respond to immunotherapy, it really depends on your initial microbiome composition. And these studies were run in parallel with a study also from my group, where we were actually looking in many different types of cancers, actually 8 different cancers, and both immunotherapy and chemotherapy. And we could also observe the same, that using the baseline microbiome, you can predict with pretty good accuracy if someone is going to respond to treatment or not. And actually, we did some mice validation also there, where we used 2 or 3 of the driver species, and we confirmed in mice that adding these microbes can really influence the therapy response. Cancer has, besides, of course, the main goal, which is to have a reduce in the tumor size and see this therapeutic effect. Cancer patients are suffering also from other problems. Almost 80% of them is having cachexia, which is basically losing a lot of weight, being nutritionally very, very weak. So we wanted to see whether the microbiome is also to some degree contributing to this phenotype. Until recently, there was no human study looking into the microbiome and cachexia. We did that. We had two groups of patients, cachectic and non-cachectic. We look into the microbiome, and again, we could classify based on the microbiome pretty well these two groups of patients. But I think our study became much more interesting in combination, if we see it in connection with a study that was published a couple of weeks later from Max Newdrop's group, which was also looking on cachexia. And what was very interesting in our study is that when we compared the microbiome of cachectic people with healthy people that were lean, the microbiome composition was very, very different. So it's not that cachectic patients have just an extreme version of the lean microbiome. They have a different microbiome. So what Max did in his study, which was a pretty logical approach, was to actually use stool from obese individuals and make a fecal transplantation to cachectic patients, assuming that this is going to restore the gut environment and actually improve the phenotype. But this was not the case. Actually, he didn't see any improvement. So it seems that in this particular case, there is much more complexity in the phenotype, and maybe we have to go to more targeted approach. And in our project, we could actually identify Prevotella copri and another lactobacillus species that were really driving this phenotype. So maybe in the future, after we test whether these are safe interventions, we can try something more targeted in cachectic patients. I'm going to change gear now and talk about metabolic diseases. Metabolic diseases have been linked also, and probably the majority of research that is out there is on the connection of metabolic diseases and the gut microbiome, especially diabetes and obesity, which is a risk factor for diabetes. I mean, this is not to be judgmental at all. It's just that epidemiological studies have shown that obesity is a risk factor for diabetes. But there is also a significant portion of people that actually have diabetes, but they are lean. And they have very different phenotype, clinical and pathological characteristics, the diabetic people that are lean compared to the ones that they are also obese. So we set up this study in order to understand whether this can be explained by the microbiome composition. So we had 4 groups. We had obese and non-obese subjects with and without type 2 diabetes. And when we characterized clinically this group, we could see that there is a very big difference in the 2 diabetic groups in insulin secretion. Actually, there is really a dysfunction of insulin secretion in the diabetic people that are lean. So we performed metagenomics, we performed serometabolomics. I'm not going to bombard you now with the data, because I think what was extremely interesting in this project is that we identified just one species, this little magician that you see in the cover of the journal that we published this study. And this magician is a bug called Akkermansia muciniphylla, AKK between friends. And actually the presence of this species and the abundance of this species related with all the indexes that are reflecting insulin secretion. So it's a very beneficial species to have. And we could actually confirm that in these germ-free mice that we used with high sucrose data, heat-killed, and also live version of this species, and prove that by just adding this one species, you can control the insulin secretion. And Akkermansia muciniphylla has been used in clinical studies before. It has been used in obese individuals that they have high cholesterol levels, but it has been used as a pasteurized. In our case, we noticed that it's important to be live. So as long as the safety tests have been performed, I think it also provides a very interesting therapeutic option for type 2 diabetes in lean people. So once again, I think we're getting closer and closer to clinical applications. Now, while diabetes is a lifelong disease, so as long as you have diabetes, you have been diagnosed with diabetes, you need to take a pill for the rest of your life. Pre-diabetes is a reversible disease, so it's much more interesting to look into pre-diabetes. And if you have been diagnosed with pre-diabetes and you go to your doctor, the first thing that she will probably tell you is that you need to exercise more. What she's not going to tell you is that 7 to 70% actually do not respond to exercise, or they even respond negatively in terms of glucose homeostasis. So there is no clinical model right now that can predict whether you are going to respond or not to exercise. It's a trial and error. So again, we came with this idea that it could be that the microbiome is a reliable predictor of responding to exercise in pre-diabetic people. So what we did in this project is that we set up a high intensity training. We recruited overweight individuals diagnosed with pre-diabetes, and we put them in 12 weeks high intensity training. And we followed them very closely with one-to-one interviews, questionnaires, dietary, and of course, in parallel, each of them had their personal trainer in order to actually complete these 12 weeks intense training in the best possible way. And at the same time, we also sampled, we had stool samples for performing metagenomics and serum for metabolomics. And now, if you look how they responded in terms of insulin sensitivity, you see exactly what the doctor would have hide from you. You see that around 70% actually responding. In this case, every line that you see here is one individual. We had in total 20 people. So the blue lines are the people that actually responded positively, and the red lines are the people that responded negatively in home IR and fasting insulin. Now, I will skip again really the bulk of the research and the details of the microbiome composition, and I will show you just a very elegant experiment that we did in mice. So what we did in mice is that we did a fecal transplantation using the stool from week 0 before they go into any exercise treatment of the responders and non-responders. And we also did a fecal transplantation in mice using the 12 weeks stool from responders and non-responders. And while we didn't observe any difference in the mice that received from week 0 stool, we see a big difference in insulin sensitivity in the group that received from responders and non-responders in 12 weeks. So it's very clear for us that exercise is actually mediating its impact through the gut microbiome, and it's shaping actually the microbiome differently in responders and non-responders. Now, if we simply look a little bit more from the functional point of view, what exercise is doing, we can see that responders have much higher levels of short-chain fatty acids and they also have a higher capacity to degrade branched chain amino acids. So this is the functional profile. Of course, how exactly exercise is changing the microbiome in different ways, it's still kind of unclear to us. What we have hypothesized is that there are some small differences in the microbiome composition at 0 week that exercise is actually making more apparent by driving the composition in 2 different directions. And that could be the result of changes again in the microenvironment, the inflammatory status in the gut, or the oxidation status. And this is how the composition is going to different directions. Now, from the translational point of view, I think the most interesting was that again, by using the microbiome and the metabolome of week 0, we could develop a machine learning algorithm that can predict who is going to respond to exercise or not with almost 90% accuracy. And we validated this in another cohort that was going through the same exactly treatment, exercise treatment in mainland China, so in another facility. So we could also confirm that this is the case. So again, of course, it has to be tested in many more people. 20 people is not enough. It has to be tested in different ethnicities, but it shows very good potential. Again, for a problem that until now there is no solution. There is no clinical model that can tell you if you are going to respond or not to exercise. I will finish with a last study. I mean, I'm coming from Leibniz Hans Knoll Institute, which is focused on infections. So I will give you a last case of, I mean, with the pandemic, we are all also more sensitive with infections. So what I was thinking for a couple of years is whether we can use the microbiome to protect us from fungal infections. We know that the gut microbiome can protect us from enteric infections. We also published a couple of papers on how the gut microbiome can protect us from Candida albicans, opportunistic pathogen in the gut. But we knew much less of how the microbiome can influence lung infections. So I focused on Aspergillus fumigatus. It's a pretty nasty infection to have. Invasive aspergillosis has a very high morbidity, so it's a very difficult to treat disease. So a couple of years ago, we sequenced 250 Aspergillus fumigatus strains, both clinical and environmental, and we learned a lot about the genomic content of these clinical and environmental strains. We just published this study a few weeks ago, but in parallel, what I was very much interested is to look into the metabolic capacities of the clinical strains of Aspergillus fumigatus. So we developed genome-scale metabolic models to try to understand more the metabolic adaptation of Aspergillus fumigatus in the lung. And at the same time, we also sequenced the sputum of 40 cystic fibrosis patients. That was a very interesting group of people because we had them before, we had samples before they are diagnosed with the infection and after. And we used another very newly developed algorithm called MAMBO in order to predict what is the metabolomic contribution of the lung bacteria before and after the infection. And if you see this figure here, I don't know if you can really see my cursor in the middle towards the right. I think it was super interesting that the growth rate, when we simulated the growth rate of Aspergillus fumigatus in the microbiome medium after the infection, the growth is much faster than before. So Aspergillus fumigatus is really shaping the microbiome in a favorable way in order to increase its growth and establish itself in the lung of these patients. So I think, again, I mean, this is still work in progress, but I think there is great potential also to develop prophylactic methods for immunocompromised patients to protect them from Aspergillus fumigatus infections. So with that, I will put a full stop here, because I don't know if I've actually already taken too much of your time. I would like to mention that we are in the process of really hiring a lot of people now, PhD students and postdocs. So if you see this type of research to fit with your interest, I will be very happy to discuss it with you. I would like to thank Novogene. Novogene has been, as Jesse said, for more than 3 years working with us. And what is magnificent with Novogene is that it's offering customized solutions. So it's not that every project is treated in the same way. We have different needs, and Novogene is always there with their expertise to hear and help us design the best way in our research. Final word is that we are organizing a really fantastic conference in Berlin next year. It's going to be a face-to-face meeting, so back to normal life, we hope, with really some fantastic speakers like Eran Segal, Karin Clement, Max Newdrop, Morten Sommer, really the pioneers in the field of microbiota. So I will be happy to see you there and maybe discuss further opportunities and possibilities. And with that, I'm done, and sorry if I took too long.
Thank you, Jenny, for your very nice presentation. It's very interesting talk. So we always know if we are on diet and if we do more exercise, we can be healthy, but we never know that this dietary and lifestyle intervention could help shaping the disease microbiome. Very interesting. Thank you for the presentation. Now let's move to the Q&A session. So the first question is that I have heard that there are so many microorganisms, but they are not all sequenced, which means that there is no reference genome at all for those unknown microbiome. But will there be a limit for the microbiome study, and how can you deal with it?
Of course, I mean, the technology is improving more and more and more, and I think it's a matter of time as we know have techniques for cell sorting, to develop techniques also from the microbiome that we do sorting really of the microbes, and then we have them by themselves in order to be able to do full genome sequencing and really get this information. Another thing that we should remember is that it's not only important who is there, but what they are doing. So more and more in our research, we are going to the direction of the functional potential, and there is of course a lot of redundancy. So even though you have a lot of microbes, there is a lot of functional redundancy of what these microbes are capable of doing. So of course there are still limitations, but I think Novogene is probably going to pioneer even that, and soon to tell us exactly the genome of every microbe that it is in the gut.
Yeah, very nice answer. Yes, Novogene could help our customers work on the de novo sequencing if there is any microbiome that you are interested.
Yes.
Okay, so the next question is there any microbial treatment using by clinician? And can you give an example?
Of course, I mean, right now we have for Clostridium difficile, for example, we have fecal transplantation, which is basically the only solution for people suffering with this really horrible disease. There is 90% success of fecal transplantation when you think that antibiotics have around 20 to 30% treatment success. Actually, when they were doing the clinical trial, and they were comparing fecal transplantation with antibiotics, they had to stop the trial because it was considered unethical not to treat everybody with fecal transplantation. So definitely, and this is just the start. There are, I think for people that are interested, they can go to clinicaltrials.org and type microbiome, and you will see that there are right now probably more than 100 studies, clinical studies that are looking how we can, with the microbiome, change many diseases, even diseases that you have not thought about it, like depression, autism. We have a lot of evidence that it's working. Where we are not yet there is to just use one species that will colonize and it will change the phenotype. But this will come as well. The most important is that we proved the causality and we understand the mechanism, how it works. The next is to bring it in the clinic.
Great, thank you. And the third question is how our genome will affect our microbiome. For example, if there are twins, they have the same genome, but do they share the same microbiome?
Yes. So the proper model, so what I have learned all these years is that there is no shortcut if you want to develop therapeutics. And that means that the clinical models that we will develop, they will use genotypic information, they will include the microbiome, they will include lifestyle choices, they will include everything. So of course, the genomic characteristics, ethnicity, we have seen that Asians and Europeans have also different microbiome. All these characteristics need to be taken into account. The model that we develop for exercise could be, until we test it, could be working only for predicting in Asians that if they are going to respond or not. We need to see if this stands true also for Caucasians or others. So definitely genotyping will be included in the clinical models.
Sure. Here is a specific question regarding to your presentation. So what is the next step for this TH17 cell paper to understand the molecular mechanisms? So yeah,
yes,
is this a single species, general, or all components contributing? Sorry.
Yeah, that's a very good question. To answer this to you, I have to say that there is one supplementary file in this paper which could have been a paper on its own to optimize this probiotic mixture for this phenotype. So it's not that we decided, let's say that one day we woke up and we said, okay, we need E. coli Nisl, we need the heat inactivated VSL, and we need this lactobacillus. Put them together in this analogy 1 to 1, and it's going to work. So we started first looking each of these different species, what can be the impact on the immune cells and on the cytokine secretion. And then after we integrated all this data, we designed this ProHeb mixture, that this mixture together is going to reshape your microbiome in a favorable way. So it's not less and it's not more than that. It is this mixture that is now on clinical trials.
Yeah, totally agree. And another question is, have you been involved in any COVID-19 studies? How about the metagenomics vs COVID-19? Very nice question.
Yes, it's a very nice question. And I think most of us, we stayed out, not because we don't believe that there will be a connection, but because everything was happening so fast with the microbiome, you knew that probably there are hundreds of groups at the same time trying the same thing. The reality is that I have not seen any paper actually really dealing with COVID-19 and microbiome. But COVID-19, because I'm looking at it from a different perspective, so I'm looking on COVID-19 and I'm trying to understand why you have responders, why some COVID-19 patients will end up in the ICU and some, they will just not even realize it that they have COVID. But we look into, actually with RNA-seq data, again with Novogene, we look into RNA-seq data from asymptomatic patients and the ones that ended up in ICU. But whenever you have such a clear distinction that you have people that are asymptomatic and you have people that really die from this disease, it really makes me think that there is something in the immune system that triggers this response. And microbiome is really a major regulator of the immune system. So I will be surprised not to see big differences between these two groups in microbiome composition. But it was also difficult to obtain stool samples from these patients. It was really, you can imagine what was going on in the hospitals with the overwhelm of the clinical sector. So they could take blood samples, but waiting when they will actually provide stool samples was almost impossible. So I think that's one of the limitations. A secret that I have is to look also into the RNA-seq data from these patients and see if I can detect from the RNA-seq data any microbiome signal. That's the closest that I will go to look into the microbiome and COVID-19. But I don't have stool samples of any COVID-19 patient.
Yeah, very interesting. Okay, so yeah, here is a question about the diabetes study. Can you comment on potential mechanisms on how echinacea affects insulin secretion?
Yes, yeah. So of course I didn't go to the details, and it's a very fair question. So Akkermansia muciniphila regulates the production of a very specific bile acid, and this bile acid is the signaling for insulin secretion. So besides putting Akkermansia in the mice, then we did the experiments in mice only with this particular bile acid, and we could see the same phenotype. So that's also another therapeutic approach, right? So one way is that you try to colonize Akkermansia in these patients, and if this is successful, then you have a natural producer of your drug in the gut and can give you what you need. If this is not a possibility, you can use this bile acid for therapeutic purposes and give to the patients this particular bile acid. Of course, it's more preferred that you don't have people always on medication, but you reshape their microbiome, you put this species that constantly can produce the bile acid that is needed. But yes, we know exactly the mechanism, we know exactly where this bile acid binds, in which protein, and how it secretes, increases the secretion of insulin. And since now this is a published study, I think you can also look into even more details if you are interested.
Thanks, Gianni, for your answer. We have another 3 questions. Yes. Okay, so the one is, what kind of machine learning models you use to predict outcomes from microbiota data?
Yes, we use different ones. It seems that the ones that are working best until now, it has been always the random forest, different versions of random forest. But we are also trying with patient similarity networks because we are trying to work also with machine learning methods that can accept zeros and or non-measured. So when you have so many patients and so many different type of data, you have the problem that you don't have all the data for every patient. You are having a lot of missing values, and the missing values and the zeros are always a problem in mathematics. So we are working now also with methods that they can accept incomplete information, and these are some other types of machine learning that are called patient similarity networks. But up to now, it seems that random forest versions of random forest are the best for the metagenomic data.
It's very
Gradient
interesting
boosting is
topic.
okay. Yeah, and gradient
Yes.
boosting, some other groups are using. I mean, it's yeah, a lot of trial and errors.
Sure. Okay, so what do you think about microbiota modulation obtained through the use of specific bacteria as therapeutic approach?
Since I'm here, I really believe on that. I totally believe on that. But for me, to be honest with you, what my research is going to be in the next years is I'm almost convinced that there is no healthy microbiome. I think the healthy microbiome is context dependent. I think the healthy microbiome really depends on the individual, n equal to 1. So what I think is actually the future is that as we go every year after the older people and we give blood for having a blood test, I will also have my microbiome sequenced every year. And the moment that my health status will change and I will be diagnosed with a disease, I will look back into my microbiome and I will see what has happened to me, exactly to me. And if I can correlate this change in my microbiome with my change in my disease status, then I know that I have to do something to reverse Yanni's microbiome back to what it was. And that's the healthy microbiome for Yanni, not necessarily for Jesse, not for John, not for everybody that it is here in this meeting. So but I totally believe that at one point I will be able to go to the supermarket and it will say with small letters that if you are missing AKK, this is the yogurt that you need. And I will take it and I will go back to my original microbiome and hopefully put my cholesterol level back to the normal levels or whatever is my problem there. But I believe it. I'm a believer.
Okay, great. And the last question, okay. What do you think about the future of microbiome modifying medication? Could probiotic treatment be a solution in mono or consortium strains, in addition to classical therapeutic therapies? Sorry.
Certainly, yes.
Yeah.
Honestly, even in the study that we did with liver cancer, I don't believe that from now on, people will be treated with probiotics instead of anti-cancer drugs. It's much more complex, but I believe that a combination could be really much more successful to regulate also your immune system or to add some bacteria that are missing before you start the therapy or in parallel. I think that this has been really shown both in human and mice studies that whether you are going to respond to cancer treatment or not has a lot to do with your microbiome composition. So a combinatorial therapy is really something that I expect to happen in the next few years.
Yeah, great. Time flies. I think that's the end of the question and answer session. Thank you, Gianni, for your very fabulous presentation and a very interesting topic.
Thank you very much. Sorry for this technical issue that I had for a second, but suddenly, yeah, everything started ringing. I apologize for that. And yeah, I hope that you enjoyed it and you enjoy the rest of the day.
Yes. Okay, then I wish everyone have a nice of the day and bye, everyone.
Bye-bye.
Bye.